All right, it's 2 o'clock, so we'll get started with our first real biochemistry lecture. So let me get the PowerPoint. Um, so I will do screen share with this PowerPoint. Let me change this. All right. As always, if you have a question, let me know via the chat. Just as a reminder, reading guides are due in the reading guide link, the turn-in link before the start of class every Monday, Wednesday, and Friday. I have not checked yet how many people turn them in, but if you haven't, since this is the start of a new semester, I will allow you to turn it in by the end of the day, so before 11:59 p.m. Without any penalties. Other than that, we will get started. So the first couple of lectures of biochemistry are a lot of review. We're going to be reviewing general chemistry, general biology, and organic chemistry. And as we move further in biochemistry, one of the things we're going to be bringing up a lot are functional groups to really understand how molecules interact with each other. We really need to see what these molecules are made of, that is, what functional groups they are. And so your first giant memorization topic for this semester, one of a couple, is to know the compound names. So you want to know the names, and you also want to know what these compounds look like for the list here and on the next page. And so the reason why I'm having you do this is that it is really beneficial and saves a lot of time when we're looking at proteins and I say, the carbonyl carbon of aspartic acid 132 is making a salt bridge with the amino group of the N-terminus alanine, alanine 1. So I don't want that to sound like gibberish forever. I want that to be understandable at some point to you. And so the only way for that to happen is for you to know these functional groups. So I guarantee you, on our first test, I will give you functional groups and I will ask you to name said functional groups, or I will give you the linkage and ask you to name them. I guarantee you, you will get some of those questions on the first test. So you might just want to put a star next to this slide and the slide that's next to it. So we will be having some questions on the test about these functional groups. All right, so I just want to see where we all are in our knowledge of general biology and some general chemistry. So I have a question here, and I'm going to have a poll to see how well we remember the videos or how well we know our general biology. Now, these polls, which should be popping up on your screen now, they will be anonymous, so don't worry, but please tell me what element in percent dry weight is the most abundant in the human body? Just to see, make sure we're all on the same page here. And moving forward, I might have enter the question music. All right, looks like we have the majority of people answering. So in the poll, share the results. So yeah, the majority of you all got that. And when you look at the dry weight of the body, we are mostly carbon. We're carbon that makes up amino acids, that makes up proteins, that makes up your DNA, makes up lipids, basically makes up everything about us. That's why in organic chemistry, you learn about carbon so much. It is critically important to us as living organisms. Now, I don't have a poll for B, the most abundant molecule in the human body. That would be water. So we are roughly 70% water. We are giant carbon water bags. That's what we are wandering around. So just to see where we are on our functional groups, see how much you remember from organic chemistry, I have another poll for you. Which atom is part, not Pratt? So that's one of my mini typos you'll see. What atom is part of the carboxyl group here? 1, 2, 3, 4, or 5. What is a carboxyl? Do you remember organic chemistry? The poll covers up the picture. So I can move it on my part. So on my screen, it doesn't cover up the picture. So you might have to try and move it on your part. So we'll give people about 15 to 30 more seconds trying to figure this one out. Minimize the poll and it won't pop up again. It might be one of your, if you click on the Zoom icon, like on your taskbar, it might pop up there or at the top of the screen, apparently. Okay. So looks like we have answers for everything, but the correct answer is 5. So that's a carboxylic acid, that's C double bond OH group. So that is the carbon part of our carboxylic acid group. And so we do have some answers for, oh, I'm sorry, share the results. We do have some answers for number 1, and that is a carbonyl group, just C double bond O, while a carboxyl group will have the OH attached to the carbonyl group. So close, but it is number 5. So again, just these slides, all of today for the most part is just really super quick refresher of organic chemistry, general biology, general chemistry to see what kind of things we'll be needing as we move forward in this class. And a question like number 2 might be one you get on tests as well, where I show you this and say carbon number 5, which group is that, for example. All right, so one of the things we're going to learn, and it's going to cover about 4 chapters in the book, if not more, is biological polymers. And this is an idea that repeats over and over and over again in life, in that life is made out of monomers, for example, amino acids, nucleotides, monosaccharides, fatty acids. And then you connect these monomers together to make a polymer, proteins, nucleic acids, polysaccharides, lipids. And these polymers are what we use to build life. So we're going to learn about the single monomers in this class, all 4 that are shown on this slide. we're gonna learn how they connect together to make their polymers. And then we're going to learn how their polymers are used inside the cell for life to happen. So this is just an example of where we're going to. But from chapter 1, what I want us to be able to recognize is, do we know the difference between these four monomers if we were able to look at them? Do you know what an amino acid versus a nucleotide versus a monosaccharide versus a fatty acid looks like? And so that's what we're going to do. So more polls. Question 3. What class of molecule is being shown here? And go poll go. What are you looking at? Is it a lipid, a carbohydrate, amino acid, or is this a nucleotide? And again, these are confidential. If you do not know, take your best educated guess. I will not make fun of you for getting it wrong, at least not to your face. And that's a joke, but no one's here to laugh, so I'm just going to assume you all thought it was hilarious. Okay. So we're waiting on a couple people, so just give it a few more seconds. Yeah, I mean, it's okay if you do not know these. That's what we're here. We're here to learn. And I'll show you what you're looking for in when looking at these molecules. What are the key characteristics to look for when you are trying to decide what these are? All right. So share results, and the majority of us got it right. And it is an amino acid. Okay. How do you know if something you are looking at is an amino acid? Okay, so an amino acid is made out of more or less, there's a major thing to look for in amino acids. It is the group I just boxed right here. So this NH3 is an amino group. This COO minus is an acid, right? It's that carboxylic acid we just looked at in the previous question. Amino plus acid equals amino acid. Now, these amino acids are going to be connected by a CH. We're going to learn about this when we get to the amino acid chapter. But just so you know, this carbon's called the carbon alpha. Anyways, to figure out if what you're looking at is an amino acid, you will always see nitrogen, carbon, carboxylic acid. Now, this nitrogen doesn't have to be charged, and this COO could have a hydrogen on it to make it neutral, but that is the characteristics of an amino acid. Always has that structure. And amino acids are what's making up your proteins. So we'll learn about that in a couple weeks. That will be our first monomer we're looking at. All right. Question 4. What is this? What molecule is this? What is super long and made out of carbon? Again, if you're not sure, feel free to take an educated guess. Just like if you were in a test situation, try to take your multiple choice answers, eliminate the ones you know for sure aren't the correct answer, and from the remaining answers, take a guess at that point. All right. 100% in. Share the results. So this is not a carbohydrate. This is a lipid. So how do we know carbohydrates from lipids? Okay, so let me get my pen back out. So for a lipid is going to be what we call aliphatic. I'm not sure if you remember that or you learned that from organic chemistry. When I say aliphatic, that means it's just carbon, hydrogen, carbon, hydrogen, carbon, hydrogen, carbon, hydrogen. That's all this molecule is made of. And then it has a little baby polar group right at the head. So if your compound is a bunch of carbon and hydrogens, and you have like a polar head group, then that's a lipid. What's a carbohydrate? Well, let's break down this, break down the word, right? Carbo, carbon, hydrate, water. A carbohydrate is sugar water, or not sugar water, I mean, it's carbon water. So when you're looking for a carbohydrate, you're looking for carbons, and each carbon, or almost every carbon, has a OH on it. They're hydrated, carbohydrate, right? So that's the difference between a carbohydrate and a lipid. Bamboozled, yes. It be like that sometimes. Just don't get tricked on the exam. I don't care if you get everything wrong here. Just when it comes to the exam, that's when it actually counts. And on the homework too, but you got 3 chances for the homework, so. All right. Let's continue our journey into what we should have learned in biology, but maybe forgot, or maybe didn't get taught it, who knows. Question 5. What is this? And go, poll, go. What are you looking at? This one might be a little tricky because it does have functional groups and molecules we talked about before, but let's see where we're at. Okay, got the majority of people in. I'm waiting on a couple, so I'll give you about 15, 20, 25 seconds. Well, I'm glad you memorized it in a different class. It makes my life a lot easier. Yeah, that is a nucleotide. So what makes up a nucleotide? Nucleotide has a couple parts to it. So let me get my pen back out. So you have your carbohydrate, your sugar. And just as a reminder, for those of you who don't remember organic chemistry, if you see solid lines mean carbon-carbon connected. So even though there's no carbon shown here, those are carbons. So you have your sugar, then you have your base, and there are 5 bases that we will memorize. If you already memorize those in a different class, you're ahead of the game. We don't need to memorize those yet, that's for further on. And our phosphates. So our phosphates are what we're going to use for energy to connect our nucleotides together and to use those for energy. But that's what you're looking for a nucleotide. You're looking for a sugar, base, and a phosphate. Oh, I forgot to share the results, but yeah, we crushed it on that one. Okay. So clear my drawings. Yay, molecular bio. Not many people say yay, molecular bio. But if you took molecular bio before this class, a lot of it's going to be the same until we get to like chapter 2. Okay, What is this? Now, let me open the poll. So, unfortunately, when you look in textbooks, when you look in papers, not everyone's going to have the nice stick figure that we've been seeing in our previous slides. This is a diagram of charge. It is often used to show molecules if you want to see specific interactions in biochemistry, because a lot of biochemistry is based on plus and minuses. So let me just, in case you're totally confused on what you're seeing here, red means negative charge, blue means positive charge, green and white's neutral. Let me just actually write the molecules. So red is oxygen, white is hydrogen, blue is nitrogen, green is carbon. So that should help. Yeah, so now that you have those, what are we looking at? Yeah, we had a question on amino acid. That was the first question. Trying to game the system like that. Not the best way to go about a test, saying, we haven't been tested on this question yet, so it must be that answer. I'm not going to just put all my questions straight out like that. That's no fun. Got to be a little bit tricky. Okay, give people about 20, 25 seconds. Your brain is crying on the first day. Buckle in. It's not too bad. I promise biochemistry is not too bad. It's just a lot of review on the front end because this is a level 4000 class. It's supposed to be tough. All right. Let's see what we have here. Majority of you got it. It is an amino acid. Remember what I told you to look for an amino acid. Nitrogen, hydrogen, 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 connected to a carbon, connected to a carboxylic acid. So there's your amino backbone right there. This representation is called ball and stick model. A lot of times when you look at papers, look at textbook, this is a much prettier way to show molecules than just the carbon skeleton. And scientists get bored after a while, and so they want to make their molecules look super pretty when they publish stuff. So this is often a way they're going to show things. But like I said, this is good in that it'll show you the charge. So this part of the amino acid is negative. This part has some positive, negative, negative, positive. And that can be helpful for us. For example, if this was a candidate for a drug molecule and we want to bind it to a specific protein, we have to know what the charges on the outside of the molecule look like to begin to ask ourselves, okay, will this actually bind to the protein? Let me look at the charges. The charges match up well, then yes, it will bind, or the charges won't match up well, no, it won't bind. But that's just what we're looking at right here. Okay. Question. Should we memorize the colors? No. Well, memorize, not so much. I got to think of the best way to put this. Okay. When we look at these colors, red being oxygen is like uniform. Like every time you see red, your brain should say oxygen. Blue is going to be nitrogen. Every time you see blue, you should think nitrogen. White is always hydrogen. So every time you see white, you should think hydrogen. Here, carbon though, carbon is usually not green. Carbon is usually black. But here, this image is having it be green. So carbon apparently can do a little bit of changing. I have never seen oxygen, nitrogen, or hydrogen be anything else. Now, the reason why I say you don't have to memorize this is because I'm not going to ask you on a test, what's this? And if I show you a molecule, like I'm not going to say, here's a red sphere, what does that mean? And if I ever show you a molecule like this, I will give you the key that red's oxygen, green's carbon. But for your own knowledge, if you ever look at one of these, that's the color code right there. So that's why it's good to know if you're going into pharmacology, if you're going into medicine, if you're going anything that deals with molecules, you're going to be seeing a lot of these. Right. Clear all drawings. One more just. Yeah. Let me get out pen mode. I believe this is our last one. Okay. Poll number 7. What molecule are we looking at here? Then I believe, if I remember right, we're moving on. And as a reminder, questions like these last ones, don't be shocked if you see on our first test, if I give you a molecule and just ask, what class is it? Hopefully, after looking at some of these, you're starting to understand what are the defining features of each type of molecule. Later on in the semester, we'll learn a lot more about them. Okay, go about another 15, 20 seconds. Okay. Yes, carbohydrate. Like we said previously, you are looking for carbons connected to OHs. We'll learn it always doesn't have to be OHs. There are different types of sugars out there. But for right now, you're looking for carbons and OHs. Doesn't have to be in a ring. Carbon or carbohydrates can also be shown in this Fisher projection. So be aware of that. Just because I didn't show a Fisher projection, if I show something like that, you should also know, oh, that's a carbohydrate as well. Again, with the Fisher projection, like in our lines here, you don't necessarily have to show the carbons. You can, nothing wrong with that, but just to save time, chemists don't show the carbons. What was the answer to 5? I have to go back to see what 5 was. And I'll be posting these answers online, by the way, after we're done with the whole PowerPoint. 5 is a nucleotide. All right, so moving on. So next, we're just going to see, do you remember general biology? So we're going to be talking about evolution in this class, but mostly it's going to be evolution of like proteins. However, we will talk about cells, and we need to know the differences between a prokaryotic and eukaryotic cell. So let me get this poll up here. Question 8, launch poll. All right, so what developed during the evolution of eukaryotic cells from prokaryotic cells? Did we make DNA, cell membrane, a nuclear membrane, ribosomes, or protein? Basically, what's the difference between a prokaryote and a eukaryote? Going all the way back to Gen Bio 1, I think. I don't know. I haven't taken Gen Bio 1 in a long time. And in this class, we're going to be jumping between an animal cell and a prokaryotic cell quite a bit, talking mostly about humans and E. coli and a little bit of yeast. You change your mind, can I choose again? I do not know if it allows you to do that, but it really doesn't matter all that much because they're anonymous, they're not graded. I just want to get a sense of where we're at. It matters to the pride. Well, if that, then write down your real answer on a piece of paper before I show it. All right. Share results. It is a nuclear membrane. Okay, so in a prokaryote, if you remember in a prokaryote, your DNA is not contained by a membrane. Your DNA is just all over the cell. It's all in your cytoplasm. In a eukaryote, though, the eukaryote has this nuclear membrane that's being shown here. So here's the nuclear membrane. That contains your DNA, and that is only found in eukaryotes, not prokaryotes. The cell membrane on the outside here, that contains all your cytoplasm. That's holding all your stuff inside the cell. So that is found in both. So it is the nuclear membrane. Yeah, so it is all membrane-bound organelles. So prokaryotes don't have membrane-bound organelles. Eukaryotes do. So yes, that is also a difference. One of many. There are a lot of differences between prokaryotes and eukaryotes, but right now we're just doing a quick review. We'll get more into differences later on in the semester when we talk about how to grow protein inside of cells. All right. Now, here, we're going to actually continue a little bit about eukaryotes. So again, this is just review. Do you remember Gen Bio? So one, where does aerobic metabolism occur in eukaryotic cells? And two, I have a bunch of statements. Do you know which one is false? Just trying to pump your memories of Gen Bio, get you ready for the journey ahead of biochemistry. Oh, and I should probably tell you what a micron is, in case you don't know. A micron is 1 × 10⁻⁶ meter. So you probably can't really picture what that means. In terms of cells, that's quite big. So that means that a eukaryotic cell is quite big. Well, after 3 to 4 years of college, I would hope you could get these biology questions. But if you don't worry. That's why we're reviewing. I know some of you probably have not taken biology in many years. Like I've had people in this class who told me, yeah, it's been a decade since I took Gen Chem. I have no idea about that stuff. And that's okay. That's what I'm here for. This PowerPoint is strictly for you to see, do I remember the stuff that I'm supposed to know? If not, maybe I should review a little bit. No worries about clicking the wrong answer. All right, so let's take a look. Aerobic metabolism happens in the mitochondria. So that is the powerhouse of the cell. That is why we need to breathe oxygen is that that oxygen is used in the electron transport chain to get that last electron. If you want to know more about the electron transport chain than you probably ever cared about, please do sign up for Biochemistry 2 in the spring, and we'll talk about metabolism for about, I don't know, 8 weeks of just straight up metabolism, but it is mitochondria. Which of the following statements about eukaryotics is not true? They are not archaea. Archaea are a whole different class of organisms. And so eukaryota are their own kingdom. So that is the false one. Eukaryotes do have organelles. Eukaryotes are massive. They are ginormous cells. You can be unicellular like a yeast or multicellular like a human. They're just not archaea. And I forgot to share the results again, but those are the results. All right. Moving on. And like I said, if you have any questions about any of this, just put it in the chat and I will answer you. And here are our 3 kingdoms, because we're going to be talking a little bit about evolution here. So you don't need to really remember, okay, what organism goes into what kingdom. I don't really care about that. I do just want you to know the 3 types of kingdoms there are. So eubacteria, archaea, eukaryota. Like I said earlier, we're going to be focusing on animals. If you came here for plant biochemistry, I hardly ever talk about that. We'll talk a little bit about that when it comes to sugars. And we'll also talk about the E. coli, which is your, it's well studied. And so mainly all of biochemistry, when you first go to learn about something, you usually use your model organism of E. coli or sometimes yeast. But that's what we're going to be looking at for our models. And just in case it's been a while since you've seen an evolutionary tree, each branch point is a common ancestor. So it's just showing you, what kind of common ancestors have branched off in the past. And it is interesting that animals are more closely related to fungi than they are to plants. Not by much, but just a little bit. That's just a fun fact we've learned through studying biochemistry and biology. But we will be talking about evolution, so let's just make sure we have the fundamentals down. Yeah, so evolution. The theory of evolution includes which of these principles? And let me pull up the poll again. Here I actually have the question in. Okay, so here are our 3 topics. Is evolution not directed towards a specific goal? Is evolution ongoing and constrained by its past? And does evolution require some sloppiness for adaptations to change? Or is it A, B, or is it all 3? And like I said earlier, we're mainly going to focus on protein evolution in this class, not like evolution of organisms, but just how proteins evolve. Sloppiness, that means mistakes. That means when copying your DNA, you have to be able to make mistakes. Mistakes must be able to happen. Yes, mutations. Okay. Share results. So yes, it is A, B, and C. So evolution, evolution is not like, well, how to put this? There is no ultimate goal for evolution. It's nice to think that humans are the be-all, end-all organism, but we are still evolving somewhat. So there is no goal. Evolution is continuously moving on based on what pressures you see in your environment. The fittest members of your population will pass on their genes and your population will evolve to meet those challenges. It is ongoing, so it is continuing to go. And you can only evolve if you have something to begin with. You are building on something. Something is being changed. You are not likely to make a brand new protein out of scratch. What's going to happen is that you're going to take an existing protein and it's going to change and it'll give you a new function. And that's what the sloppiness means. You need an accidental mutation to happen, right? So usually, actually always when it comes to evolution, let's say there's some new food source available for bacteria, and one bacteria is able to eat that food and the others aren't. That's because that bacteria didn't copy its genome correctly. It had an error. It was a beneficial error, a beneficial mutation that allows it to get a new food source. But that only happened because it could not replicate its genome perfectly. And so that is also a requirement for evolution. You have to be able to make mistakes. Those mistakes must be passed on to future generations. So A, B, and C. For adaptation, it's not, so adaptation to change isn't goal oriented. It's just that it can change and sometimes it gets lucky and makes the right change. So for example, you can put E. coli into a very unfavorable environment and they are 99.9% going to die. For example, you don't give them a food source that they need. You don't give them an amino acid that they need to survive. So we'll actually look at this in Biochem too. But what will happen is that the E. coli will turn on what's called the SOS mechanism, in that it knows it's dying in a couple minutes or an hour. It can feel that all the systems are going bad. So what it will do is that it will turn off a lot of its genome proof editing and just say, genome, I am not going to correct you. We're going to die anyways. Start making some mutations for me. And maybe 1 out of a billion cell will make the right mutation so that that cell can survive. Now we have a population that can survive these harsh environments. So it's not a goal. They weren't mutating to try and for a specific problem. It's just a random mutation that may be beneficial. It's just very hard to do. So that's why it takes a lot of E. coli. Yes, we have no sympathy for E. coli in biochemistry. We do such horrible things to E. coli. But I mean, you kill E. coli every time you wash your hands, so you shouldn't feel too bad about that. Don't use soap if you want to save the E. coli, I guess. This is a pandemic, so I shouldn't say that. Use soap to wash your hands, everybody. All right. The last thing we're going to talk about is from general chemistry, everybody's favorite topic, thermodynamics. I know I had some of you in Gen Chem, and I'm sure when I got to the whole idea of thermodynamics, you were just like, yes, I love this. Well, congratulations, it's back. So let's just start here. I'm going to go over this slide, and we'll probably have to save the actual problems for next week. Okay, so this is your major equation. Delta G equals delta H minus T delta S. Delta G, Gibbs free energy. This will tell you if any reaction in the universe will happen or not. So spontaneous means will happen. Whoa, my pen's not working. Okay, spontaneous means will happen, right? So if delta G, and I actually have it over here. If delta G is negative, a process will happen. If delta G is positive, a process won't happen. If delta G is 0, you're in perfect equilibrium. All right. Delta G is made out of 2 parts, H and S. Enthalpy, I do not know how to spell. Enthalpy and entropy. They sound very similar. H is enthalpy, S is entropy. H basically means heat. S basically means chaos or disorder. All right. These signs can be positive, negative, or zero. So if a process has negative enthalpy, it's exothermic. Exo, think of it like exiting. If you're exothermic, heat is exiting your body, which is good if you want a reaction to happen. If it's endothermic, delta H is positive. Heat is going inside your system, and in. So heat's going inside your system. Not good if you want a reaction to be spontaneous. S, positive. Chaos increases. Very good. If you were to actually look at this equation, the Gibbs free energy equation, and go to understand how these terms are derived, you will see nothing in the universe happens without chaos of the universe increasing. So the only thing anything happens in the universe is if somewhere in the universe is becoming more chaotic. So delta S, chaos increasing, good. If you're making a reaction more ordered, chaos is decreasing. That's not good when it comes to spontaneous, but that can be balanced out with heat. So that's your basic thermodynamic equation. Throughout biochem, we will see delta G a lot, and I mean a lot. Just remember, negative delta G, spontaneous, will happen. Positive delta G, non-spontaneous, will not happen. And we're talking about thermodynamics, and what you need to remember about thermodynamics or energy is that energy and kinetics are separate topics. So just because you know something will happen does not mean you know how fast something will happen. Take a metal or iron nail and put it next to the beach. You know it will rust. That's spontaneous. It's just going to take a while for it to rust, so it'll be slow. Positive delta G, bad, because nothing happens. If you want the reaction to happen, yes, it is not good. I don't know if I would say bad, because it depends on what you want to happen. For example, well, what do I want to give an example for that? Okay, the positive delta, so the reaction of me of flying off the earth spontaneously has a positive delta G. It won't happen. I would claim that's good because I don't want to go into space without a suit. So it depends on how you ask the question, but it means positive delta G won't happen, negative will happen, zero, you're at equilibrium. Let's see here. Yeah, so that's about all we have today. Let me just see what our next question is. Okay, we will have this one quick poll question, and then I'll call it a day. And on Wednesday, we'll get into math, everyone's favorite subject, I know. But I just want to make sure we understand the idea of entropy or chaos. So we're going to do this one quick poll question, and then I'll let you go. So what has greater entropy or chaos? Liquid water at 1 centigrade or ice, or 0.1 centigrade, or ice at 0. And for that process shown below, is entropy increasing or decreasing? Remember, every time you see the word entropy or you see delta S, I want you to think of the word chaos because it means the same thing. So what has more chaos? Liquid water or ice? what has more chaos from that reaction? And since we're more or less out of time, I'll just go about 20-ish seconds. Then I'll tell you the answer, and then we'll call it a day. All right, so let's do this really quick. All right, so water does have the greater entropy, more chaos, because water is moving around. The atoms in water are free to move in any direction. Ice, the atoms can't move. They can vibrate, but they can't move in any direction. So liquid water is a lot more chaotic, a lot more disordered than ice. For the reaction, you're taking two gas molecules that are free to move in any direction, and you're combining them into one gas molecule. When you take gas and you, actually you're taking four gas molecules and making them into two, but anyways, when you decrease the amount of gas molecules in a reaction, entropy is decreasing, because they are, the more pieces you have, the more chaotic it is, right? It's almost like Legos. What's more chaotic? 4 Legos spread across the room or 2 Legos? 4 Legos are more chaotic than 2 Legos. So yeah, that is all we have time for today. Wednesday, what we're going to pick up is doing some free energy calculations, and then we will go from there. So if you have any questions, let me know. I will post a homework up on Blackboard, and I'll tell you how to get to that. I'll make an announcement from that. But if you have any questions about anything we did, please do send me an email. Otherwise, I'll see some of you Tuesday for lab, and I'll see hopefully the rest of you Wednesday. Have a good rest of your day.